Hello, today we are going to talk about normalizing the table of expression. RNA-seq data has been shown to have skewed distributions, unequal variances, and extreme values. Most statistical tests, including ones that we use in our analysis, have an assumption of normality. The three aforementioned issues violate these assumptions. There are a number of normalization approaches that may be used. A standard approach includes conversion of values to log scale and adjusting the distribution to account for technical variability of low expressed genes, aligning the mean of distribution between all samples in a set. On the T-Bioinfo platform, both of these can be done using the Quantile Normalization utility. This utility can be found on the main areas of analysis page. Quantile normalization and other normalization procedures are important to transform distributions both between and amongst different samples to have the same distribution. This removes unwanted technical variation that could cause perceived differences between samples when there are in fact no differences in terms of the biology. Quantile normalization is a technique of mapping original values to the corresponding quantile of a standard distribution. Here, an estimation of the sequencing depth of an experiment is determined by an upper quartile of its counts. Comparing quantile values instead of raw values will eliminate differences solely rooted in the technical variation, such as the amount of RNA sequenced, sequencing machine type, or even unexplained differences that rise between different technicians following the same protocol. In addition, while normalizing data, we will use a log transformation that makes it easier to interpret differences in gene expression. To use both normalization and log normal transformation of the expression data, select Quantile Normalization after Start and set the following settings. The transform table to ln should be yes and threshold should be 5. Now we are ready to download the table output and compare expression between FPKM and normalized data. A good way to compare normalized and adjusted data is to take a few samples and visualize all gene expression numbers as a box plot. This method of data visualization, popular to summarize data, shows the maximum, minimum, and mean values for each sample, while displaying the interquartile ranges of upper and lower interquartile ranges. Thus, we can see some detail but not too much to lose the trend. You can prepare the box plots in Excel to see how it can show us a comparison between FPKM output and normalized data. The first figure shows the majority of values that is not seen at all because there is a gap between the majority low levels and some outlier high values in the selected samples. After normalization, this gap disappears, placing most of the data into a more easily distinguishable scale. In fact, it shows the variance of data that is needed for a comprehensive comparison. For example, if we prepare a box plot of all the samples, we will see that some of them are outliers. The outlier status of the sample is not by level of expression of any single gene. It shows that the variance of this dataset is different from the others. The mean of the sample is just a little higher than the rest, but the distance between the mean value of the lower interquartile range and the maximum of the top interquartile range is much bigger than the rest of the samples. To learn more about normalization, you can visit lesson number 12 under transcriptomics on learn.omicslogic.com portal.